Hello and welcome to our discussion today on Crohn's and colitis, microbiome biomarkers, diet, and the environment, hosted by sister companies, DNA Genotek and Diversigen. With inflammatory bowel disease impacting over 3 million individuals in the USA alone, today we wanted to bring our attention to the outstanding ways in which researchers are exploring how to leverage the gut microbiome and its biomarkers in developing new, less invasive diagnostic tools and different therapeutic, lifestyle, and dietary interventions, paving the way to advanced understanding and the future of a better quality of life for these patients. I'm Andrea Hardy, registered dietitian, here to moderate this incredible panel of 3 experts. Please ask your questions in the chat as we start to explore a multi-omic approach to gain a deeper understanding of disease occurrence and the development of novel therapeutics in IBD. This webinar is being recorded and everyone will receive a follow-up email with a link to the recording shortly after the event. Let's meet our speakers. Our first speaker is Gerard Honing, who is the Associate Director of Research Innovation at the Crohn's and Colitis Foundation. His particular focus is on venture, philanthropy, and precision medicine R&D to advance products with the potential for positive impacts for patients. Gerard received his PhD in neuroscience at the University of California in San Francisco as a fellow of the Howard Hughes Medical Institute and subsequently trained in immunology and microbiome research. In 2013, Gerard developed and implemented a novel clinical stage therapeutic modality for the treatment of C. difficile infection. This experience led Gerard to co-found Symbiotic Health Inc., a biotech company focused on the development of novel drug delivery systems. Gerard serves on numerous boards and committees, including the board of directors of the Peggy Lillis Foundation, project teams for the Foundation for the NIH, the Health Research Alliance Program Committee, and the Mentorship Committee for the Entrepreneurship Lab NYC. Thank you for joining us today, Gerard. We also have Dr. Allison Zhang, who is a postdoctoral scholar at Stanford University. Dr. Zhang joined Michael Snyder's group in 2018 at Stanford University. Her research focuses on understanding the exposome, particularly how the exposome affects human health. She has studied the human exposome in multiple environmental settings, including hospitals and during wildfires. She recently launched a study to look at how environmental factors affect the onset and flares of Crohn's disease using exposometer and multi-omic profiling. And then lastly, we have Dr. Kevin Litkowski, who is the Director of Molecular Technologies at Seres Therapeutics. Dr. Litkowski graduated from the University of Pennsylvania in 2006 with a Bachelor of Science in Bioengineering. And in 2012, he received his PhD from Boston University in Biomedical Engineering. He is currently the Director of Molecular Technologies at Seres Therapeutics, where he has been for more than 8 years and has led the creation and growth of the Molecular Technologies Group to develop, execute, and understand molecular approaches to characterizing microbes and the microbiome to support all of Seres' programs through preclinical and clinical development. Thank you guys so much for joining us today, and I will hand it over to Gerard first to talk a little bit about what IBD is and how it relates to his research.
Sure. Thanks everyone for joining. Such a pleasure to be here, and thanks so much to DNA Genotek and Diversigen for the invitation. I'm really delighted and honored to be on the panel. So Crohn's disease and ulcerative colitis collectively are inflammatory bowel diseases. These are inflammatory diseases that impact the gastrointestinal tract primarily, although not exclusively the gastrointestinal tract. They also can have an autoimmune component, particularly for Crohn's disease. Crohn's disease and ulcerative colitis have a number of distinguishing features, but primarily have to do with the location of disease. Ulcerative colitis principally affecting, although not exclusively, but principally affecting the lower gastrointestinal tract, colon and rectum. Crohn's disease can affect any part of the gastrointestinal tract, anywhere from the mouth to the anus. Inflammatory bowel diseases are are associated with very significant impact on morbidity within the US population, affecting likely millions of individuals within the United States and also worldwide. There's, of course, an important link between the onset of inflammatory diseases and a variety of factors, including genetic factors, environmental factors, particularly diet, as we've seen with westernizing diets and lifestyles associated with increases in IBD in the developed world and also in the developing world increasingly. And we also, of course, have learned a great deal over the recent decades about the relationship between gut bacteria and actually not just gut bacteria, but gut bacteria of the microbiome and inflammatory bowel disease pathogenesis and disease activity as well. And so, as Andrea mentioned, I'm here representing the Crohn's and Colitis Foundation, which is a nonprofit organization which was founded in 1967 with the mission to accelerate towards cures for inflammatory bowel diseases and to alleviate suffering caused by those diseases. And in connection with that mission, the organization has had a longstanding interest and active investment in a variety of areas of microbiome research, including, I think, some of the topics we're going to discuss today. And of course, I personally have been working in the microbiome field for about 10 years, and I'm really incredibly delighted to see the incredible advances that my colleagues on the academic side, like Allison, and also on the industry side, like Kevin, are rapidly advancing forward. So yeah, I'm delighted to be here. Thank you.
Thank you, Gerard. I will pass it off to Allison to tell us a little bit about her research.
Thank you, Andrea, and thank you all for joining this incredible webinar. I'm really great pleasure to be here. So so now people realize that environmental factors also play a role in the onset of IBD. So the exposome, which is a rather new concept, is introduced by Christopher Wilde in 2005, and it refers to all the exposures across one's lifetime. So this not only includes those physical exposures that you're exposed to air, water, soil, heat, UV light, and this also involves like socioeconomic factors, lifestyle, educational level, and also your psychological status like stress and anxiety. So there are lots of research showing that the exposure plays a role in IBD, and this can be as early as when you are born. Like how you are born, whether it's natural birth or C-section, they both play a role in your risk for IBD. And also if you are breastfed or have you got any antibiotic exposures in your early life. And later in your life, your lifestyle, of course, your diet plays a huge role in the IBD pathogenesis. And also if you are smoking and whether you are stressed or anxiety status, these all plays an important role for the onset of IBD.
Great, thank you so much, Allison. And then we'll have Kevin talk a little bit about his work with ulcerative colitis.
Yeah, thank you very much, and thank you for having me. I joined Seres in 2012, close to its inception, and we've been working to develop microbiome therapeutics in a number of different disease indications. We currently have 2 therapeutics in development for ulcerative colitis, and I'd like to just give a brief overview of those and describe my role in them. Seres is a biologically sourced product, which means we purify the firmicute bacteria from stool and formulate it for delivery. We believe that this allows us to focus on the bacteria we believe most responsible for a beneficial effect on disease. It allows us to inactivate and clear pathogens and optimize the pharmaceutical properties. We tested this in a phase 1 study where we saw favorable safety results and very encouraging, statistically significant results in clinical remission in the vancomycin and daily dosing arm. More details of the study can be found in Gastroenterology Journal online and soon to be in print. And we're currently testing Seres in a phase 2 study. One of the ways in which we used what we learned from that study was to help inform the design of our next candidate, SER-301. SER-301 is a rationally designed therapeutic candidate for ulcerative colitis. where we were able to select strains for the composition by developing the design criteria based upon these learnings, and then testing the candidate strains in vitro and in vivo assays. In particular, we were able to learn about strains, species that engrafted well in patients, and species and microbially derived metabolites that associate with remission. This allowed us to develop a composition that we think can target multiple mechanisms and pathways that we had seen targeted in measures of patient biopsies from that SER-287 study. My role in this work is to help understand how we generate this sort of data that can inform these learnings, and how we understand the limits of that data collection. So in close collaboration with partners such as Diversigen, we've worked for years now to characterize our assays, starting all the way with sample collection and storage, and understanding the impact that that has on the results and conclusions that we can draw from these assays. We have also done a lot of work in trying to maximize what we can learn from the patients that are participating in these studies and from each sample. And so that's where we've been able to look at ways in which we're able to do multiple assays for each type of sample that we get, using some of DNA Genotek's solutions, for example, to broaden the settings in which we can try to get these samples and the types of assays that we can run on them. And we're really excited and looking forward to seeing how we can continue to apply these learnings to our existing candidates and broaden our portfolio so that we can bring next generations of microbiome therapeutics to patients.
That's fantastic. Thank you guys for that brief summary of what you're doing and how you're utilizing this incredible data. Let's jump into the Q&A. So any participant that has questions, please be sure to pop it in the Q&A section, and I will be sure to be asking those to the panelists. And I'd like to start with a question of my own, if that's okay. I'm going to direct this towards Gerard. Could we talk a little bit about some of the research you're participating in with the Crohn's and Colitis Foundation, specifically around consolidating data across cohorts, and how doing that can help to generate new insights and increase the impact of IBD research?
Yeah, absolutely. So the Crohn's and Colitis Foundation initiated targeted funding directed at the microbiome research field in about 2008. And at that time, a lot of that funding actually went towards the academic laboratories at the Broad Institute and other institutions that were developing some of the computational tools that were really important in terms of making biological sense of microbiome-derived data. And we're very fortunate to be involved in some of that research that has borne fruit in terms of enabling research in the subsequent decade. And now, looking to the future, we are in an incredibly dynamic and productive time within microbiome research, and I think we've seen the kind of amount and quality of data increase dramatically with decreased cost of sequencing, increased focus of many researchers on this topic, and also the advent now of a number of clinical stage therapeutics companies and many other preclinical stage companies focused on microbiome-based therapeutics and other technologies. So that is all for the best, but one of the kind of chronic challenges within the space has been the lack of sufficiently powered clinical cohort studies in order to, so that we're going to talk about a number of the different factors that can affect the microbiome, but fundamentally it's a highly dynamic organ, which is responding to many different inputs, including diet, a variety of medications, particularly but not restricted to antibiotics, many other exposures, genetics, etc. So that's part of the power and promise, right? This is a mutable organ that can change over the course of one's life and in a variety of states in health and disease. But that's also an enormous challenge when you're looking at typically cross-sectional studies of relatively small numbers of patients, perhaps a few dozen, and then crunching microbiome-based data can be quite overwhelming to understand really what are the most critical signals and which are the specific clinical or biological factors within those patients that are related to particular patterns which are seen within the microbiome, right? This has been a chronic challenge. So the foundation's kind of one of the active phases of microbiome research currently, it has to do with combining multiple different sources of data and also different types of biosamples with very rich clinical metadata from thousands of patients from multiple cohorts, and making that resource available to our colleagues on the industry side and also on the academic side. And so that's the philosophy of our data exchange and biorepository platform, which is called IBD Plexus, which at this point has data from tens of thousands of IBD patients. And this is a combination of many different types of data, including stool samples, and at this point, very rich set of molecular data, whole genome sequencing, and also as well as 16S from multiple thousands of patients with both ulcerative colitis and Crohn's disease, both pediatric and adult. And so this, and one of the kind of critical aspects of this effort has to do with enabling larger cohort studies by first recruiting, of course, large numbers of diverse patients that are truly representative of the patient population in order to allow for reasonable numbers of important but small subgroups, such as highly vulnerable patients, patients with aggressive disease, patients with treatment refractory disease, patients with complicated inflammatory bowel disease, which we can get into as well. So all these are very important, and over the lifetime of a patient with IBD, they may enter into one of these highly vulnerable situations, but to study them has been historically very challenging. So IBD Plexus has a number of different components, including very large observational cohorts, which are followed over many years. So for example, our pediatric cohort is now in its 10th year of observation and still has a few hundred patients who are actively being followed. This will be the last year for that study. And this is something which is very difficult for individual academic organizations or drug companies to support, this kind of very long-term follow-up. But that's something that we're focused on doing as a nonprofit that's really has this long-term commitment to the disease space. Then another aspect, which is where our collaboration with the Diversigen comes in, has to do with utilizing core laboratories and standardized protocols for sample collection, processing, data generation, as well as data processing, in order to reduce or at least minimize the types of experimental variation that typically is observed when 10 samples are run in one lab, 20 samples are run in another lab. And so this is one of the tools that we have, among others, in order to attempt to build cohorts. And of course, we're just one of the organizations working on this. But in order to build cohorts where you have really rich longitudinal data coupled with very high quality microbiome data and other omics data linked within the same patients with follow-up over multiple years, and some standardization with respect to experimental protocols and data generation, which we see as one of the important enabling tools for our colleagues who are exploring this incredibly rich area of research right now.
That's great. Thank you for describing that. I think it's really exciting to be able to expand and find more meaning in the data by way of having these large cohorts. So that's fantastic. And I know maybe Kevin can speak towards some of those limitations in comparison with clinical trials, because that's really where his research lies.
Yeah, there's I think a lot of similarities, right? The key points around standardized methods, I think, are really valuable and really are an asset for the field to be able to draw conclusions across studies. The clinical studies, I think, do provide a different perspective. I think there's a ton of benefits that Gerard just described for these really large open studies, but clinical studies can be more controlled. have more strict eligibility and exclusion criteria to better define the patient population. And then we get to look at an intervention and, look at our drug compared to placebo and really see the differences, both in what's happening with the drug and what's happening in response to the course of disease and hopefully improvement in that. And so it gives us a different perspective on the disease because of just the nature of the study. We also have sort of the whole quality realm, where all of the assays and data is very strictly controlled, and again allows us to make again maybe a different set of claims, but with a lot of weight behind those, hopefully.
Great, thank you for sharing that aspect of research with us. And while you're on, could you touch a little bit more on how your team is leveraging microbiome in the development of new drugs and determining efficacy of those next generation designed drugs?
Yeah, the biomarkers are a critical area for us, both for informing design, finding, sort of broadening the scope of biomarkers to include the whole microbiome really opens up a very rich, complex data set to allow us to look for these biomarkers of patients that are, disease, the severity of the disease, how likely we think they are to respond to treatment, and then hopefully biomarkers that indicate successful treatment. So we're able to measure those in our clinical cohorts as part of our studies, use those to inform decisions about moving those forward, relate them to our preclinical in vivo models where possible. Again, that's not always translatable, but I think one of the ways in which we're able to use those biomarkers is to best understand in what ways those models are applicable and translatable. And then, as I mentioned, try to use some of these findings to inform the design by optimizing the compositions to target those biomarkers that we think are most important.
Right, so it sounds like in that preclinical data, you were looking at determining which biomarkers are going to be most applicable and moving forward. Then in that, you said you're moving to phase 2 trials with the SER-287. Do you have particular primary outcome markers that relate to those microbiome markers at all set for that particular trial?
Yeah. We are looking at obviously the safety and efficacy of the drug as endpoints for the study, and we are looking at a number of different biomarkers as part of that study. And we'll probably be able to speak more about that study once it's wrapped up.
Absolutely. And just in regards to one of the participant questions, he was hoping you could touch a little bit on the phase 1 trial, how many patients were in it, and whether it was mild, moderate, or severe disease as well.
Yeah, our phase 1 study had sorry, I want to say it was 58 subjects, but I can double check that. And it was in mild to moderate patients. They were randomized across 4 arms, 1 placebo group, and 3 different treatment arms in the early study to look at different dosing regimens. As I mentioned, there's a lot more details available in the paper and in the supplementary methods. It details all of the, I believe it details all the inclusion and exclusion criteria for that patient population. So a whole lot of information available there.
Right. Yeah, definitely read the study. I learned a ton from it. So thank you. So to all of the speakers, maybe we can have a bit of discussion around this, but how does gut microbiome testing help us to look towards changes in diagnosis and treatment? Kevin, I think you highlighted well some of that drug development pathway. Maybe the other speakers can speak a little bit to other avenues for diagnosis and treatment as well.
Should I start?
I'll pick on you. Yeah.
Okay, so the prospects for the question is related to one, diagnosis, and then selection of appropriate treatment. That's the focus. Okay. So for currently diagnosis of IBD is based on a constellation of different assessments. This includes clinical presentation, endoscopy, a variety of laboratory tests as well, and also histopathology. The laboratory tests are typically looking at relatively well-established inflammatory markers in the blood and in the stool. No one test is particularly sensitive or specific for inflammatory bowel disease. In fact, diagnosing inflammatory bowel disease is non-trivial. In general, distinguishing it from irritable bowel syndrome, which is often comorbid with IBD, is also a challenge. So there is certainly a need for a range of different biomarkers in order to address multiple different aspects of inflammatory bowel disease. These include diagnosis. So this would be identifying inflammatory bowel disease and correctly characterizing someone as having inflammatory bowel disease in a patient who is suspected or symptomatic. And also distinguishing inflammatory bowel disease from other conditions such as irritable bowel syndrome, which has a very, very different treatment trajectory. So this is one challenge, and then identification of the appropriate treatment pathway. So we're fortunate in the inflammatory bowel disease field to have significant amount of innovation in recent decades related to the introduction of multiple different approved therapies. I believe there are 8 in the United States currently. And of course, there's also surgery, which is for Crohn's disease, for example, approximately 50% of patients over their lifetime will experience one surgery currently. Hopefully that will decrease, as the research progresses. So diagnosis is one. Then there's question of monitoring the disease over time, identifying whether treatment is working, and also selection of appropriate therapy. In terms of diagnosis, there are certainly marked changes in the gut microbiome, which are present actually relatively early in disease. And those have been characterized both in pediatric Crohn's disease, also in ulcerative colitis as well. To date, though, none of those tests have exhibited the performance characteristics that would be required in order to supplant current standard of care methods for diagnosing IBD. And probably they never will. I mean, an endoscopy is there's always going to be a reason to do it at least once or twice in an IBD patient for a variety of important clinical reasons. So that said, certainly there is the prospect of adapting the types of microbiome analyses that have been done in inflammatory bowel disease in order to come up with molecular classification tools that could aid in the diagnosis of IBD, and eventually potentially also in monitoring disease activity and identifying the appropriate treatment. And so that would be kind of characterized as diagnostic biomarkers, monitoring biomarkers, or prognostic biomarkers, which can indicate which patients are likely to experience severe or aggressive disease course or complicated disease course, such as formation of strictures, which are basically blockage and scarification of the intestine. And then the kind of potentially most impactful of these would be biomarkers that could in advance predict which patients are most likely to respond to a particular therapy. And so there have been multiple publications on all of those different aspects. And the notion that the microbiome analysis would completely supplant traditional methods of assessment, I think, is not realistic and frankly, maybe not desirable. But in each of these areas, the potential to utilize the microbiome to move beyond traditional methods of classifying patients in order to come up with more objective and quantitative methods of assessing each of these different aspects of of inflammatory bowel disease is a highly promising area of investigation. And I do, however, want to mention that a number of the currently clinically used biomarkers, this is, I don't think necessarily something that's well recognized, but a number of the clinically used biomarkers include, for example, immunoreactivity to commensal microbiome organisms, which ordinary people, healthy people shouldn't have. Patients with Crohn's disease often have, and the presence of those antibodies can be helpful for diagnosis and also for prognosis. Even fecal calprotectin, which is detected in the stool as an inflammatory marker, is effectively an antimicrobial peptide, which is secreted by neutrophils in response to presumably invasion of microbiome bacteria into places of the gut where it shouldn't be. So we're already using biological factors that are really very closely tied to the microbiome clinically currently, and really the sky's the limit for the potential for microbiome-based biomarkers to move forward and improve patient care.
Yeah, it's very exciting to kind of see how these pieces of information will round out our clinical picture, our ability to do research, and then our ability to diagnose and treat these patients. Somebody in the chat also had a little bit of an add-on to this question, in that they're wondering if there's any tests available or that we've looked into for people with a parent who have Crohn's or colitis to see if there's specific genes or microbiome markers that would put them at higher risk of developing IBD. Is there anything going on like that in the research or in the work you're doing?
So certainly there are a number of different genetic factors which have already been identified. Oftentimes these are genes that are involved in innate immune response to microbiome factors, some of which are incorporated into combinatorial clinical tests. And studying first-degree relatives of patients with inflammatory bowel disease has been an active area of research. There is not currently any susceptibility. What you're talking about would be susceptibility. There are no clinically available tests that would aid in identifying in a healthy person what their risk is of inflammatory bowel disease. And there's a variety of reasons why that research is highly challenging, practically speaking. But it is not out of the question that could be developed in the future. But as of now, it's a very active area of research, but it is not something which is translated to the clinic today.
Thank you. So let's shift gears. Allison, I'd love to touch a little bit on your research. In particular, I'd like to know, how can exploring the metagenomics of the human exposome give us better data in understanding how environment plays a role in disease, as opposed to just solely looking at the environment?
Sure. So traditionally, when we look at environmental research, we use data collected by weather stations, and that has a lot of limitations. Number one is the information that a weather station can give. They give you the total PM concentration, they give you the concentration of a handful of gases, and also like temperature and humidity data. But out of all those particular matter concentration, you never know what's actually in there. So for the same concentration of particulate matter, it could be different based on the effect. The effect could be different based on what's actually in there. And also the weather stations are located in a let's say one bigger geographical area. But from our previous research, we found out that even people with the same general geographic location, their airborne exposure could be different. And this, so by looking at the exposure at the individual level instead of just the general environment level, we can get a more individualized profiling of the exposure. So just for the area exposure, for example, we use a wearable device to collect your daily exposures, and we use a multi-omic approach. So for the biological agents in the air, we use next generation sequencing. This will be metagenomic sequencing instead of 16S, just to give you an idea of what kind of microbes you're exposed to. And also there are also chemicals in there, and there are also interactions between the chemicals and microbes. So we use mass spectrometry to analyze the chemicals in the air. And more importantly, we not only just want to characterize the external exposures, we want to learn more about how those external exposures affect our health. What does it change? How do those exposures change your internal? So to profile your internal status, there are a couple of ways. First, of course, for IBD patient, gut microbiome will be on top of the head. And also we use some Multi-Omics research. And so far we're only using blood metabolome, but this could be also done with for gut microbes and also proteome. And we want to associate how the external exposures affect the human health by correlating the external exposure with the internal environment.
That's great, thank you. And so it might be a bit early in your research, but we have a question from Jennifer Leuenberger, who is wondering which environmental factors are most prominently associated with flares in IBD. Are you able to answer that yet based on where your research is at?
Yeah, to be honest, our research is still early in this process, but from previous literature, like smoking and diet is one of the top triggers that could cause flares.
Yeah, that
Yep.
absolutely.
And,
oh, go ahead.
Sorry. Yeah, diet is a big one. Unfortunately, the specific dietary triggers, I think, as Allison was talking about, that's what still not quite unraveled. It's highly individual, and so understanding that relationship is important. The other is environmental. The other environmental factor, which has certainly been shown to increase flaring, not just in IBD, but in a number of chronic inflammatory conditions, is stress, psychological stress, perceived psychological stress. It's another big driver.
Right, and would you say that any of the current research is looking at gut microbiome markers and metabolomics in diet or stress at this point? I think diet, yes, but maybe you can speak a bit to that, towards that.
Sure. So yeah, so environmental factors and the role in inflammatory bowel disease is definitely a major focus for our funding efforts, and we have multiple programs ongoing in that regard, including a number of randomized controlled trials and also observational studies related to dietary triggers and potential dietary triggers of disease activity. We've certainly been very interested in, as Allison mentioned, the role of smoking and other environmental exposures. And then in terms of psychological stress, that is another area which we are very interested in, and so we're supporting two studies. One at University of Alabama and one at UCLA. In one case, looking at children who with inflammatory bowel disease who have experienced significant trauma, early life trauma, and looking at the relationship in those patients as well as in animal models of early life trauma between inflammatory markers, vascular function, and the microbiome. And then at UCLA, we're following up on actually a significant history within the irritable bowel syndrome field, looking at relationships between stress sensitivity, and basically stress reactivity within inflammatory bowel disease patients, flaring, and basically relationships between gut microbiome and functional magnetic resonance imaging, for example. So, all very early and exploratory work, but the general concept that there is a relationship between psychological stress and diet and the microbiome, and that those have some significant relevance for the lived experience of inflammatory bowel disease patients, I think is reasonably well established as a conceptual framework.
Great, and tied to that question, we have an audience question. that I'll just modify slightly from Williams-Turpin in regards to evidence that the microbiome is involved in IBD onset. So case control studies have observed a difference in the microbiome, but could this be just a simple reflection of established inflammation or dietary habits, or do we believe that the gut microbiome drives IBD onset as well?
So I can start. So this is sort of a fundamental issue with many cross-sectional studies in inflammatory bowel disease patients, where you basically identify differences in the microbiome between patients with established IBD and healthy controls, which is patients with inflammatory bowel disease have chronic inflammation, dietary changes, variety of things that could actually drive microbiome changes, where those microbiome changes were not causal. I think some of the evidence suggesting actually that the microbiome is driving disease relatively early have to do with the fact that, first of all, significant microbiome changes can be observed in patients at the time of diagnosis. And so this is presumably before they've experienced, multiple years of inflammation, A. And then B, those same microbiome factors that can be observed early in disease can also drive colitis in appropriate animal models. And the animal model question has been kind of very difficult to develop over time, but at this point I think there's quite a few convincing models that basically if you can manage to transplant these some of these bacteria which are observed very early in disease into the right animal model, they can drive inflammation, whereas other bacteria will not. But I'm curious to hear Kevin's thought about the causality of the microbiome in this regard.
Yeah, we've I think focused a lot on the causality in treatment rather than in how the disease arises for obvious reasons. And we do think that there's a causal role. That's sort of the basis for our therapeutics. And isolating that is one of the areas in which having these interventional studies, whether they're our own clinical studies or other FMT studies, really help get at the causal nature of the microbiome in these diseases and allow you to tease that apart more finely. I think the point about the animal models is a good one. I think you always have to focus on which aspects of those models reflect which parts of the disease, right? With such a multifaceted disease, right? Many of our models reflect parts of the disease course, and again, focusing on that and understanding which parts of that are then translatable when we think about the therapeutic development is really, I think, the key question in that space. But I definitely agree that there's a lot of progress there, even if you're sort of looking at a mosaic rather than one single picture.
Great, thank you. And so a question to all speakers. So how do you incorporate, you kind of all addressed this, but the start of the question is, how do you incorporate multi-omics and multi-biomarkers in your work? And more importantly, how do you decide the sequence of investigation when studying the mechanism? So any tips or things that you guys consider in your research that you can pass along to fellow researchers?
For us, it's the questions of sample availability, right? What do you have? What can you work with? And then really, what are your questions and what are your capabilities? I think I saw a question there on the how do you then make sense of all of this data once you generate it, which is a key piece of this research, and it's an area where we've focused on in building our research engine. Is investments in characterizing, again, not just the sample collection and data generation methods that I mentioned, but the data analysis methods, developing our reference data sets, and really tuning them to the sort of space that you're looking for. There are a lot of great resources out there, but we feel that we've been able to build, leveraging our strain collection, we've been able to add to those databases and help us see more of the bacteria in the spaces that we're looking for, and then link them up with the the other omics that we're looking at, whether it's metabolites or otherwise.
Yeah, we you know, we're all really primarily about enabling other organizations to further their research. So we basically go with the community consensus. And I will say that when we do collect multiple different types of samples, dual samples are always the first to go. And so in terms of prioritizing, one thing I would say to the group is if you're focused on, you know, basic research of how the microbiome functions and the basic biology, that's great. If you're principally interested in impact on patients, you need to start with the clinical problem that is you're attempting to solve, really laser focus on that, and really start with the patient's experience and the biology of the patients at that, you know, at that particular time, and kind of work really from there. And sometimes that'll take you in the direction of the microbiome, sometimes not. But, you know, really thinking about, you know, what is the ultimate solution that you're attempting to deliver in the long run, and you know, whether you're starting from a firm foundation, firm data and biosample foundation in order to address that gap.
Great. Oh.
Yeah, sorry. Just to add on to that, it's just so for multi-omic approach is basically to find out what is the one that most interests you. For us, we of course want to use exposome and correlate exposome with everything else. For others, maybe gut microbiome is the top hit, and maybe metabolome is the top hit. So this is basically using your top hit and then go with others. And also depends on what kind of applications of your research. If you are just interested in the general course of Crohn's disease, probably just looking at which omic best reflect the change of disease, or if you are interested in eventually providing treatment or diagnosis for the IBD patients, you want to find out the OM that's most related to the health outcome of those patients.
Great, that's definitely something to consider. And on that note, I think this leads really nicely into discussing some of the limitations or challenges we might face using metagenomics and microbiome biomarkers for IBD research. Is there things that the audience should be considering in terms of the limitations there?
Yeah, I think we've touched on a couple of them already in terms of reference data sets, in terms of, I think there have been some improvements, but there's continued work on standardizing methods and being able to compare that data across studies. And then I think the biggest one is still the complexity of the data. And the need to really invest in being able to understand it. Omics provides, you know, a wealth of data, which is tremendously informative, but there's a lot there. And then particularly when you look at as diverse of a patient population, and again, I think what Gerard just mentioned about understanding the disease, right? Which question are we asking? How targeted is that question? And you'll be able to get, at least ask more targeted questions of your omic data. But I continue to think that the complexity, you know, is both the value and the challenge.
Yeah, I agree completely. A few other issues that come up again and again is have to do with sample collection and where the sample is being collected from. Obviously, stool has a lot of advantages in terms of not requiring a procedure, the extent to which it reflects it's really reflecting principally the biology of the cecum and the colon, maybe not so much upper GI tract. So that's always an issue in terms of where the sample is coming from. characterization of the patient and understanding of the many factors that can impact the microbiome, which one is really driving the changes. One of the issues which can come up is the fact that you can see changes in the microbiome that are affecting very common microorganisms. Those may or may not be the ones that are driving pathology. So you may have relatively numerically uncommon bacteria that have a very important function, biological function. For example, these are some of the challenges. And I think in terms of practical application, the practical challenges with reducing eventually the complexity of the assay towards something that can be implemented more routinely, I think is something that the field, as it advances, is going to have to face in order to bring some of these technologies to the clinic.
Yeah, just to add on that, the multi-omic data is definitely very complex, and it's really hard to interpret. And one thing that we really want to be cautious about is driving causal or correlation. So lots of the data come out of a multi-omic research is correlated, but a lot of them are not causative to IBD. So when drawing conclusions, we got to really be cautious about determining whether something is causative or not.
Great. Yeah, I think that's really important to highlight for people. Well, guys, this brings us already to the end of our panel. There were so many great questions. I know we definitely did not get to them all, but hopefully that allows us to be able to run an excellent panel in the future where we can get to some of those more those other great questions. So I want to say a big thank you to all of our panelists, and thank you guys so much for joining us today for this incredible discussion on microbiomarkers. If you guys have specific questions for the participants, you can grab their email off of this slide. For those of you that are interested in using DNA Genotech microbiome kits and Diversigen's service offerings, please be sure to visit the links here and get in touch for services and trial kits. If you are interested in learning more about microbiome research and studies in multi-omics, be sure to check out their educational blogs and their other webinars like this, and DNA Genotech's white paper on Omnigene gut for sampling in IBD patients. As Gerard, sorry, can we, next slide, thank you. So as Gerard had mentioned, the Crohn's and Colitis Foundation, it has access to those incredible tools, including IBD Plexus, and they have issued an international request for proposals for academic researchers to gain access to those IBD Plexus resources. So if you are interested in having the opportunity to gain access to the data and biosamples collected from the ontimalinib, clinical trials, you can submit a letter of intent by December 31st by visiting this website. And for Allison's research, if you are interested in participating or know somebody that's interested in participating in Allison's research on the exposome in Crohn's, they are looking to recruit participants, and you can contact them for more information at crohnsexposome@list.stanford.edu to learn more. And then to learn more about Kevin's research, we had talked a lot about that new paper in gastroenterology. So be sure to check out that phase 1 trial with Seres 287. To learn more about their approach and pipeline, visit serestherapeutics.com. And if you want to participate or know somebody that wants to participate in their ulcerative colitis study, you can visit eco-resetstudy.com. A follow-up email with this information and a recording of today's webinar will be sent out later in the week. And thank you so much for attending, and I hope you guys all have a really great day.